So, all right, we're going to talk about V1. I actually changed the title to this more biblical title. The Yeah, God, that's gonna interrupt me. People keep coming in. I hate that. I stopped paying attention to that in a minute. All right. So first, let's talk about the eye and the how the signal is transferred from the eye to the visual cortex, to the early visual cortex. So I'm sure you all know the physics of the eye. There is the lens. You have the image on the retina, and when you zoom on retina, retina has this strange structure. And if a wise engineer had designed it, he or she would have designed it in a better way. But this is evolution, so sort of stuff and tricks stacked up in evolution. So something is basically upside down in retina. If you look at the retina, the photoreceptor layer, the layer of neurons that are photosensitive, they absorb the light and they create the neural signal. This is the first front of our sensory system interacting with the physical world. That is in the back of the retina. That's in the deepest layer, the outmost layer of the retina. So the light has to go through all this junk that is between the photoreceptors and the lens to make it to the photoreceptors, activate a photoreceptor. Then that activation pattern propagates back in two layers. There is this layer of neurons called bipolar cells that you see here in this very dark green. They get the signal from the receptors, photoreceptors, the blue, green, and red stuff that you see here in this drawing. They activate the bipolar cells, and the bipolar cells activate the ganglion cells. And there are neurons in between too. There are horizontal cells between the bipolar cells and photoreceptor cells. Also, oh, you can see my cursor now, right? Yay! So one thing has improved. So these are the photoreceptors, right? The signal, the light goes through these cells, hit them. The light gets distorted, of course. The quality goes down, and then they activate the bipolar cells. Bipolar cells activate the ganglion cells, and the ganglion cell axons gather together, travel on the retina, and exit the eyeball in a point that we call optic disc, or that's where the optic nerve exits the eye, and there's a hole in the eye there. There's no photoreceptor. To the left, there is a drawing of retina by Santiago Ramon y Cajal, one of my favorite scientists of the 2 centuries ago. And as you see, it's pretty much the same as now. That's why I like this drawing. It's very accurate. And now the back of the eye is on the top. The light is going from the bottom as if you're looking down. And on the back of the eye, you get the rods and cones, the photoreceptors, then the bipolar cells, then the ganglion cells. And that's an electron microscope picture of same thing. So the reason I am bringing this up is to remind you that processing of visual information starts from the eye itself. From an embryologic point of view, retina is part of the nervous system, part of the CNS that has grown into the eye, and processing starts there. If we did not have those 3 layers of neurons in the retina to process the signal, those are the Mexican hats and stuff like that, right? They're compressing the signal. If we did not have that, the width of the optic nerve had to be much thicker, because you would need to do that computation in the brain. The other point is that what I am going to talk about today is mostly about the main central function of vision, the basic functions of vision. There are lots of things that happen in the retina, and we are ignoring them, and they are very interesting things. This is evolution, right? So yes, evolution happened to be like go this way. The photoreceptors are back there, and light has to go through all the other neurons, communication neurons, and they are not photosensitive. But now that's the case, let's evolve some photosensitive ganglions or photosensitive bipolar cells. That has been shown that there are types of, let's say, ganglion cells or bipolar cells that actually have photosensitivity, and they respond to light. And that has been a puzzle. What do they do? Samer Hattar here at NIH has done amazing work on that. He has discovered some of them that are sensitive to light. They see light, but they are not for vision. They are for the circadian rhythm, controlling your sleep cycles. And so there is a lot to be said about retina that we are skipping. Retina looks like that. If you look at it from, oh, somebody is coming in. If you look at it from an ophthalmoscope, you look into the eye, that's what you see. There is this tree of blood vessels, and to make the things worse, actually, in terms of design, the blood vessels are also on the way for the light, so they cast a shadow on the retina, actually, and you don't see your blood vessels because that shadow is always in one spot in your retina, and the system has adapted to it. But this is another experiment I thought we could do together, like the mirror experiment, but that's a little awkward, so I'll just tell you, and you can go do it afterwards whenever you want. You would need a bright, thin flashlight and a dark room. So you would look into the infinity, into like sort of a far away in a dark space, close one eye, and shine light into the white of your eyes, not into the cornea. Like look away and shine the light into the white of the eye and move it. That light will go through your eyeball. It will illuminate the inside of the eyeball, not through the normal path, but through the wall. That will move the shadow of the blood vessels on your retina, and suddenly you see the world cracked. You see you can see all the shadows of the blood vessels. That pattern that I am showing you, you will see it if you start moving that flashlight into your eyes, and then you will feel blind for a few minutes. So you see where the blood vessels converge, that's the output of the eyeball. That's where the blood vessels and the neural elements, basically the axons of the ganglion cells, exit the eye. There is a hole there, so you don't see things there. That is the blind spot. The center of that image, you see a little bit of darker. There is one point which is dark. That is called macula or fovea centralis. That's the center of your fovea. That's where you have the highest density of cones and the best visual resolution. And as you see, there aren't many blood vessels occluding that. All right, so okay. Now, let's do a little experiment with each other. I'm sure you have seen it before. Close your left eye and look at the red dot with your right eye. And move your head back and forth until you don't see the black circle, the black disc to your right.
How far from the screen should we be?
It depends on the size of your screen, but
I'm this far and it works.
Right? Yes, Simon, I guess you should be farther.
Oh, okay.
It works for me like that. I'm kind of 1 yard away, something like that. Can everybody disappear, the black dot?
Yes.
Yeah.
Let's make sure that everybody got that, because we are going to play with it. Yep. Is there anyone who has not disappeared the disk? Anybody? Right? Okay. So I want you now to to look at the dot, look at the red dot from the distance that the black dot is now on your optic disc in your blind spot, and you don't see, you see like a flat white screen. Is everybody there? Keep your eyes on the dot, on the red dot, keep it, and keep looking. All right. So now you should be seeing a uniform stripe pattern, vertical, coming in, filling in that hole. There is no hole in this prism bars. Now go and check the disk is still there. Make an eye movement to the black disk. Go back to the red. Go back to the red dot. Make it disappear. Make it disappear. Look at the red dot. Let me see. Okay, it's gone for me. Now your field is filled with a set of horizontal gratings, not vertical ones. Make an eye movement to the disk. Make sure it is still there. The black disk is still there. Go back to the red dot. Go back to the red dot. Make it disappear. Now your visual field is filled in, and actually keep your eyes on the red dot. I can fill it in with an image of the world, and you don't see a hole in that. Now you can go and check the black dot. So, and what you see is that when you do not see things that are in your blind spot, but your brain is filling the world for you, is filling in the gap. And that's another one of those things that I mentioned. The vision is a constructed experience. So basically there is this hole in your visual field that you don't see it. And how does it fill it up? With the background information. So what happens from there? So when you go outside the eye, there are these two cables called optic nerve. Each one of them has 1 million wires in it, 1 million axons in it. Check the numbers. We have about 100 million photoreceptors, 120 million photoreceptors. So that's in the order of magnitude of 100 million. By the time the signal is exiting the eye, it is 1 million. So those processings, the Mexican hats, the edge detection in the retina has compressed the signal by a factor of 2. That's a lot of compression. That means that your optic nerve had to be 100 times thicker, like a hose coming out of your eye, if you did not do that processing. The two optic nerves go and cross each other at a place that is called optic chiasma. Then they separate off and make this thing called optic tract and enter thalamus of the brain. So thalamus, so this is the optic nerve, that is the chiasma, this is the optic tract. This goes inside the brain to thalamus, lateral geniculate nucleus of thalamus, and it makes a synapse there. From there, it projects through what we call optic radiation, because it looks like a fan, into the visual cortex in the back of your eyes, in the back of your heads. So if you have had the experience like me of falling down and hitting the back of your eye or your head with something solid, you can remember that the first thing you experience is not pain, it's light. You see a flash, and that's because your V1 is being mechanically stimulated. In that diagram that I showed, and that's the standard classical diagram, lots of important things are missing, are ignored. So this is another way of looking at same thing. There are two cables, optic nerves coming out of the eyes. They cross at the optic chiasma, and I talk about that crossing. We have to understand how does that work. They go to the thalamus. Well, one branch of them goes from the thalamus, goes to the cortex. But if you look at these collateral lines in this photograph, like here, what is that? That's an output of thalamus. Well, that actually crosses the thalamus, and that does not go to the cortex. That goes to superior colliculus. Superior colliculus is part of your brain stem. It's a very ancient architecture in the brain. And that pathway is very important. That's how snakes see, because a snake, a reptilian, does not have cortex. So that's basically the old visual pathway that the reptilians, the birds, they see with that structure. In the mammalians, the cortical pathway have taken over, although even in mammalians, if you go to rodents, still most of the input to the most of the retinal output goes to the brain stem, does not go to the cortex. So this heavy loading of cortex with visual information is to some degree a primate thing. You see it in other mammals too, depending on the type of mammal, like is it a visual mammalian or not. But in primates, most of the output from the retina ends up in the cortex.
Arash,
But we should,
I have a
please.
question. So I've seen I can't remember what the type of blindness is called, but there are patients who don't have conscious vision, but they can do tasks like navigate a cluttered hallway, or
Yes.
take an envelope and put it in a slot. Is it the
Yes.
case that their cortex is what's damaged and they're relying on superior colliculus, or is there something we actually don't know what's damaged?
It's a very good question. And can you hold it? Because I have a separate slide for that case. That's called blind sight. And we'll talk about that. But thank you for bringing this up, because those collateral pathways remain untouched when visual cortex is damaged, and they contribute to visual function. Also, as you see, some of them go to other nuclei in the brain stem, and they are responsible for your sleep cycle, things like that. So let's ignore that for a second, ignore that evolutionary thing, and we are more interested in the primate visual system. So let's talk about that. The way the crossing happens is something that any student of vision should be very familiar with, because that's your bread and butter, in a sense, to calculate what happens when you show something in the visual field. If you see this image, we have painted the left side of the visual field with red and the right side of the visual field with cyan, just to encode the information better. We have two lenses, right? And the lenses make the image, flip the image, make it upside down and flip it left, right. So on the back of your eyes, the left visual field is presented on the right side of the eyeball, and the right visual field is presented in the left side of the eyeball, right? Now, your retina is split in terms of wiring. Your retina, the left side of your both retinas are going to see the right side of the world, right? And the right side of your both retinas are going to see the left side of the world. As you see marked here in red, like that part of the retina, you see my cursor, right? Cursor, yep. So that part of that part of the retina is seeing this part of the world, right? This other eye also sees that part of the world. These two go to the chiasma, and a very interesting magic happens at chiasma. The fibers that are going from the nose side of the eye, from both eyes, they cross. The fibers that are coming from the ear side of the eyes, from both sides, don't cross. As a result, the red lines from both eyes end up in the same visual tract, optic tract. And the cyan parts of both eyes end up in this other side of the and basically, as a result, your left hemisphere sees the right of the world, and your right hemisphere sees the left of the world. This is a very common mistake, that you would say left hemisphere, right hemisphere, left eye, right eye. If you want to isolate one hemisphere, should you close one eye? No, because each eye is connected to both hemispheres. So if you want to isolate a hemisphere, you should isolate half of the visual field, actually, right? The easy way of thinking about it is that if you look at the tip of your finger, everything from that point that you are fixating to the left is now in your right hemisphere. from both eyes. Everything that is from that point to the right, from both eyes, end up in your left hemisphere, right?
Why, so
Now,
why is that advantageous to have both sides on both sides?
beautiful question. Beautiful question. If you go to the evolution and evolutionary past, you see this very interesting effect. The animals that are more like prey animals, the animals like basically the herbivores, the animals that are being hunted by the meat eaters, they usually have their eyes on the two sides of their head. Like think about a rabbit, the eyes are here and here. Basically, they want to optimize and maximize the size of their visual field, right? Because if a predator can attack from anywhere. So they want to have big visual field. And in those guys, the two visual fields do not overlap. So you have one eye covering here, one eye covering here. And in the chiasma, the cross section is complete. So basically, you don't have that funny wiring. Each eye goes to a hemisphere. When you have the animals, mostly predator animals, mostly meat eater animals, that have their eyes, they have their eyes in front of their skull. They have overlapping visual fields because using that information, they can solve the problem of 3D vision, and they can accurately position things in 3 dimensions. If you are a predator, if you're a cat, that matters a lot because you only have one shot at that bird that you want to catch, right? So you have to perfectly calculate your distance and attack. But now, so you have to have overlapping visual fields. But as soon as you have overlapping visual fields to support your stereo vision, you have got another problem. Same thing is being captured by two eyes. So now, do you want to let's say my hand is in one part of your visual field, two eyes. Do you want to put two different resources in your brain to deal with that same thing, or you prefer to project that stimulus into one spot in your brain? That is basically the logic of this wiring. So when you have the overlapping visual fields, you want to make sure that same things in the world go into a same spot in the cortex. Is that clear?
Yeah, that's cool.
Yeah, Arash, I have two brief questions just in case you.
Please.
So the first one is like the figure you have there shows a clear overlap there, like in central vision of. So when at what distance are these kind of things really separated by hemifields from the perspective?
There you are. Sorry, say that again. I was looking for you.
In the image you have here, so if the point where you're converging is very close to you, there's an overlap between both eyes. So there's a central field that is kind of you're looking at with both eyes.
Yes.
So my question is, like, at what distance do you know approximately what distance this thing is already there independent?
Independent, you mean that the angles look the same?
So that the left hemifield goes to the right hemisphere and the right hemifield goes to the left, because there's an overlap. So where does I guess they have to be like folks fixated at some point. How far would that be? Like a meter, 2 meters, or?
I will be talking about that extensively at the stereo vision session, because basically what you are asking is a quite important geometric question on how the geometry of the world represents to the short version is that there is a circle called horopter, and the horopter is a circle that is passing through the two eyes and the target, and that determines the circle. And the geometry becomes funny for things that are inside that circle and things that are outside that circle. Basically, the way they represent themselves in the back of the eye. And we have a total separate session for that because that's more of that's an elaborate discussion actually.
Okay, cool. I'm gonna be looking forward for the horopter.
Yeah. So everybody got this concept, right?
Sorry, Arash, it's Amanda. Hi, how's it going? Could I also ask a question about the evolutionary advantageous of how eyes work. So it was a great question about like predatorial versus predator. But my question is like, what does pupil size have to do with it? Because I know typically like predators, we think of like big cats have that very elongated, almost like football shaped predator eyes,
Right.
and they allow for like a certain amount of light. So what's the difference between like predators and prey allowing light to hit the retina? Like what, can you just add that to your description of it so I can understand?
That's actually a very good question, because preys usually have horizontal
Yeah.
pupils, and predators have vertical. Not all of them, though. Not all of them. That's that. Well, for the prey, you want to maximize the field, right?
Okay.
That's what you want to do. So you want to maximize the visual field around you, and to do that, you would need to basically, it's like a slit. The wider it is, the better.
I
It
see.
gives you a wider visual field.
Mm-hmm.
But when you want to solve the problem of 3D vision, that's a curse, because now you have to find corresponding spots, and if you have a horizontally elongated image, it will be, most of it is useless basically for you for solving the correspondence problem. And if you remember that, we'll talk more about that in the stereo vision session.
Okay,
It's actually
thank you.
interesting that questions about that.
Does,
Had I known this, I would have maybe I should have done the stereo vision before this.
maybe
Simon.
this is a question for the future, but does the shape of a pupil affect the field of vision?
A little. You know, like theoretically it shouldn't, because it's a lens, right? So theoretically it shouldn't affect it, but it is very close to the lens. It's an aperture that is close to the lens, and yeah, it does. It ends up. I mean, at the end of the day, oh, people are still coming in. At the end of the day, I got confused. What was it?
Just the shape. It was based on Amanda's question about the kind of the horizontal or round pupils versus like the big cats with their kind of oval pupils. It's weird.
No,
they are different.
And actually, it's a very interesting topic. It's not classical physics. How the light diffracts through when you change the shape of the pupil. The most interesting case of that, and I just mention it briefly and move over because that we have another session on that, is the case of color for octopus. Octopus is great in color vision, right? They can mimic, like wherever they are, they camouflage, they become like the colors around them. So they are detecting colors. They are pretty good behaviorally for discriminating colors, but they don't have color sensors. They have only one type of cone. They don't have the 3 cones, RGBs, whatever, short, medium, long wavelength cones that we have. And how do they do that? It turns out that the shape of, if you look up octopus pupil, they have a very like horseshoe shape, very strange shape of pupil. And that is to diffract the light. It functions like a prism. And so that on the eye, you get this diffraction pattern. Some parts of the eye only see the cyan part of the image. Some part of them only see the red part of the image. So now you can have a labeled line. You can say, whatever activates this spot on my retina is red, and use that to reconstruct color. It's pretty amazing and very different from what we do. So let's do before moving on from this topic, because I want to make sure that you have completely grasped the notion. Let's go through a practice that every medical student, second-year medical student, go through it, and that is when you know the wiring diagram of the visual system. If you have deficits in vision, by the properties of the deficit, you can tell where is the lesion in the brain. Like you have a patient with an infarction. We don't know where the infarction is, and you are in a village far out, whatever. You want to know where in the brain that has happened. By village, I meant you don't have a fancy imaging facility around you, let's say. So how do you tell where is the lesion by measuring the behavioral impact of the lesion? So let's do it through, let's use this diagram from American Academy of Ophthalmology to do this practice. So you have the left visual field and right visual field, right? Now you're the doctor, you go get your patient, close one eye and test, like, do you see my hand here? Yes. Do you see my hand here? Yes. My hand here? Yes, 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 yes. Okay, left eye is fine. You make a drawing, left eye is fine. And then you close the left eye. Do you see my hand? No. Do you see my hand? No. Here, here. No, 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 no. You end up being, oh, look at that. That's the top row on the left here. You see that the left eye is seeing, right eye is blind. Where is the lesion? Can somebody say where is the lesion for that? Loud.
It's literally labeled there.
Yeah. It's the optic
It's
nerve.
the optic
in the
nerve,
right
right?
eye.
Yeah. So when the optic nerve is gone, one eye goes, the other eye. And so let's say I've lost my right eye. Do I see both sides of my visual field? Yes, of course, right? Because this eye sees both of the visual fields. Now, let's say the cut is here. Let's explore this case. The cut is right at the center of optic chiasma. What happens to vision? Somebody explains.
Well, the nasal part doesn't cross. So the projections to the nasal retinas don't cross. So therefore, in the contralateral side, you don't see that portion.
So basically what you see is that when you cut the crossing fibers, right, there you're cutting the crossing fibers, and those are the nasal fibers, right? So basically now if you cut that, what was crossing from here? That was crossing from here, right? What is that seeing? That part of the world. From this eye, what was crossing? This was crossing. What is that seeing? That part of the world. So what you see is that look at option 3, in both eyes, the visual field is gone for the lateral part of the visual field, right? So in this eye, I don't see here, I see here, that's fine. In this eye, I don't see here, here is fine, right? So my visual field.
It's like looking like in a tunnel.
It's like narrowing down. Yeah, it's your visual field narrows down from the sides. It's not a tunnel because the top and bottom remain the same, but left and right squeeze into each other. And that's a case, actually, you as somebody who has taken the vision course, learn it right now. I have had cases in my life that actually, like, you see somebody and they tell you about this and you diagnose them. That is a place that you see hypophyse stem tumors, cancers of the base of the skull happen there, and sometimes the cancer, it remains undetected, and the only thing that happens is that the patient's visual field is getting narrower and narrower and narrower. And not as a doctor, as an observer, as a friend, as a family member, when you see that happening, you should send that patient right away to the doctor, because that could be cancer. That could be hypophyse cancer eating the optic chiasma. It's very close to the optic chiasma. So now,
about that.
What was that, Tim?
I just have a question about the kind of damage to that crossing, I guess. So you're saying that when it's getting damaged over time, the visual field decreases rather than like fragments on the peripheral. Right? And why is that? Why is it not like, the right half of my right visual field is getting fragmented?
Blurry.
Yeah, blurry rather than kind of closing in.
Good question. So these are cases that you are cutting on a cable, right? Imagine you have a CCD camera and you are basically cutting through the cable. Once you lose the cable, you lose the part that is represented by the cable. The thing is that we have geometric proximity in these cables. Basically, the cables that come from the same spot are next to each other. So when you make a localized cut, you're cutting the cables that are representing spots that are very similar, very close to each other, adjacent to each other. And that's why you get these whole areas getting lost. However, if you go at the boundary of the lost vision, like if you actually go and measure, where is my cursor here? Like that boundary, if you measure it carefully, it's not a sharp boundary. It's kind of like what you described. It's a spotty. But when you make, like, when you chop half of that cable, optic nerve, the points that you lose are not randomly distributed in the visual field. They usually come from same spot. So now let's say there is an infarction happening here. What will that do to the visual field? Somebody explains. I want one of the postbacks to explain this.
Come on, guys.
You lose the nerves from both of the right sides of the eye, of the retina. So right?
Correct, exactly.
So the left
exactly
visual field.
Exactly, you lose a whole visual field, right? Now you lose a whole visual field. Now you don't lose an eye. This is because this is after the split. That dance of wires has happened, and now everything from left visual field is there. So from that point onwards, you start losing half field. So in both eyes, right? That's number 4, that here in both eyes, half of the visual field is gone, right? And that remains the case for anything from that point upwards, right? The only thing is that from that point, the higher up you go, the smaller the lesion becomes. Because that's the choke point. All of the vision is going through that optic tract. But once you go to the cortex, it fans out, it gets bigger. And if you have a damage that actually takes all of that, it usually causes lots of other things too. So if you're explaining with a small local damage, you usually get a portion of the visual field gone. If the damage is higher up, is actually in the cortex. But if you are here, you lose like half of the visual fields in both eyes. All right. So this is from a J Neuroscience paper by Adams and Horton. Very interesting study. They have blocked the input from one eye to the cortex. And there is this staining, CO staining. I don't want to talk about that. We don't have time for that. You're welcome to check the paper if you like. But the idea is that the neurons that keep firing and keep responding to stimuli for the time that is after closure of the signal from one eye, they will be stained separately, differently in the cortex. So to the top left here, you see the retina of the monkey, a squirrel monkey that was used for this study. To the right, you see the V1 cortex of that monkey stained with CO staining, and you see interesting things. Look at that. The shadow of the optic disc. That is the shadow of the optic disc that you can actually detect on the cortex. And the blood vessels of the retina. Do you remember I told you that they cast a shadow and the brain takes away the effect of that shadow? You see them here. You see them here. So they are casting actually an anatomical, they are projecting to actual neurons in the visual cortex, and you can reconstruct the pattern of blood vessels of the retina from V1 cortex. So that's basically what this study has done, and they have found the exact correspondence of that blood vessel with that part of the V1, and they call these angioscotomas. It means that scotomas, blind spots that are induced by the shadow of the blood vessels over time, over long time, and the histology should do something about that to fill it in. That's quite interesting, right? You did not see that disc in your visual field. The visual field, Reza.
Why do we need an optic disc in V1? I mean, why the cortex has something corresponding to this?
Why? Why? That's exactly the question, right? We expected the optic disc to be gone by the time you get to the cortex.
I mean, there is no optic disc input, so why V1 has something corresponding to this?
Exactly, right? There is no input there. It is made up. The thing is that the vision is a geometric sense, right? So to respect that geometry, you need to fill that gap with something. And it is a very interesting question.
But
we don't know.
Synapses, I mean, the ganglion synapse in thalamus. And then,
yeah.
I mean, it's at least one or two synapses far from the photoreceptors.
Right.
So why we still need some gap between the, I mean.
If I'm understanding Arash's answer about the geometry, the idea is that if you didn't have that there, the field would be distorted, right? It would be like you pull, if you have a hole in the V1 represents the visual field retinotopically, and you want to maintain that structure. And so if you didn't just put in a filler there, like if you had an Excel spreadsheet and you had some empty cells, instead of writing like NaN, if you deleted them, it would screw up all of your rows and columns if you just deleted those 4 empty cells. So instead you write NaN or.
But we have cortical magnification. We have, I mean, different part of retina has different representation on V1.
Right.
Exactly.
That least must be minimized to be, if not zero, almost close to zero.
Right. Yeah, that's strange. Actually, it is. The problem is that actually they use this property to calculate the cortical magnification better than anyone else, because now we have the corresponding points on the retina, and it remains a puzzle. Why do we have a piece of cortex, basically used for representing a part of visual field that doesn't have any input? Right. So my first reaction is probably it has something to do with consistency and geometry along the lines that Rosa mentioned, that the cost would be distortion of the visual field. You could imagine that something down the V1 would deal with that. Why V1? Maybe because you need to fill it up with the next door textures. What are those neurons responding to? Feedbacks too. Yeah, they actually studying that is a very interesting topic and a very hard topic, because basically putting an electrode there and recording from the blind spot of retina is a quite exciting topic actually.
And that's just to make sure I understood correctly. So
Fernando.
Yeah, the point that is being projected there is the point where the optic nerve sort of leaves the retina.
Yes.
That's where you have a high density of blood vessels. That means that this is a location where you have very low level of receptors, if not
zero receptors.
So the cortical magnification area is very far from that. So that location is not close to the area of cortical magnification. Quite the contrary.
It's not that big. Yeah, it's a small.
So it's far. The fovea, it would be like far away from that location.
Yeah.
So
check here.
Then comes the issue of the geometry. You want to. So if you have local dynamics and you have low frequency sensitivity, like apparently it happens in visual cortex in areas that are peripheral, then it's like a pond, like a swimming pool, isn't it? So if you have low frequency stuff and you leave the place that is missing, you can sort of fill it in by the dynamics of the region, no? So
I don't know.
It would allow you to fill in with a meaningful thing in an area that is sensitive to low frequency.
Right.
Does that make sense?
I guess that's the idea. Yeah.
Okay.
Yeah.
Arash,
that's the idea.
And what do you see in LGN? Because LGN is also retinotopic. You see, like, what does that region of LGN look like?
I don't think we know.
LGN has, I mean, it's not like LGN, cortex. It has different layers, and
LGN is like an onion. It is layered. It's 6 layers, and so 4 layers are magnocellular, 2 are parvocellular, and I don't think anybody knows actually what happens in LGN here? I don't think anybody knows. So actually, I'm bringing this up to mention even V1 is an area which is far from understood. There are things. Somebody wants to say something?
I can save it for the end.
Okay. Okay. So they actually went ahead and calculated what was brought up here, cortical magnification factor. If you go back to this picture, you see here, this is V1 cortex. That is fovea. Fovea is a very small point in the retina, is a teeny tiny point in the retina, but it is quite large in V1 cortex because it is packed with lots of neurons. This part, these areas are basically taking, this is like a huge part of the cortex, which is taking pretty much the same space. Not the same space, it's still bigger than fovea, but proportionally less space. From that, you can reconstruct how the world looks like on V1 cortex, right? So if that's the right visual field, the left visual cortex will represent the world kind of like that. That is the type of distortion that you are experiencing. You're seeing the world through that now, actually, at the moment. And that's, again, interesting because the size of this thing remains the same when I move it around, but on your cortex, it shrinks as I move it to the periphery. So you need to basically be correcting for this thing called cortical magnification. In vision experiments, we calculate carefully what area of the cortex we are stimulating based on where we are in the visual field. So before moving on from this study, there is another thing to notice here, monocular crescent. What is that? That part. That represents the part of the eye that the other eye doesn't really reach. So we are front eye kind of creatures, right? Our eyes are in the front of our skull, but they don't fully overlap. There is this little bit of far lateral part of my visual field which is not represented by the other eye. So that is the monocular input to the cortex. And that defines these other stuff that we see. What are those stripes? So if you remember, I told you this study, they blocked the input from one eye, right? So those lines are called monocular columns. Basically, when you go into V1, because this is the part I told you that the goal is to have the image from one part of the visual field represented on one bit of cortex from both eyes. But how do you deal with the wiring of that? Wires are coming from left eye for this point in my visual field. Wires are coming from right eye from this point in visual field. They end up in V1 cortex. In V1, they are interacting with each other, but they are still separate. Each one of those cells are representing only, if you record from that point, the cells there represent only one eye, left visual field from the left eye. Next door, left visual field from the right eye. And they are next to each other, right? If you remember, they are supposed to go to the same point from both eyes to the same point in the cortex, and that's where it happens. After V1, we don't have monocular columns anymore. They converge into same neurons. So when you go to V2, there is no eye distinction anymore. Eye distinction is gone. So by looking at the activity of a high-level neuron, let's say an IT neuron or even a V2 neuron, you cannot tell if it is coming from this eye or that eye. And would you know this? Did you know this, that even you yourself cannot tell which eye is the source of an image? Actually, if you do it psychophysically very carefully, if you look through a stereoscope and I flash one point in one of your eyes and ask you which eye was it, you can't tell. You're at chance because you're reading the activity of those binocular cells down the road that has merged the visual field, right? And you don't have access to activity of your V1, conscious access. That's a question for later. Mirror test. Get your mirrors out. Everybody. Now I want to see everybody. All right. Get your mirrors out. Perfect. So now what I want you to do for this experiment is to get your mirror. It's ironic that I tested this on myself last night, but I forgot to bring my mirror. So I pretend I have a mirror with my phone. This is the mirror. This is the mirror part of your mirror, right? Put the mirror on your nose and tilt it a little bit to cover your left eye. Not cover, but just to block the path of your left eye to this image, right? Hold it in a way that with your right eye, you can see the monkey holding the mirror. With the left eye, you don't see the monkey. You see the mirror, right? Now, get your hand out and move your hand in a way to place the picture of your hand on the face of that monkey. Just make them aligned. And if you want it to work well, move your hand out. Ideally, the distance of your hands to your mirror should be the same as the distance of the monitor to your eye, so that you don't run into focus problems. So get your hand on the monkey face and start shaking, moving your hand, waving your hand. The monkey, parts of the monkey should disappear in this weird moment that suddenly, it's very hard to get rid of the eyes. Eyes are such a strong stimuli. But you, oh wow, I get this scene that there are two floating eyes in the air and the rest of the monkey is gone. Who said that? Who saw that?
I saw that. I saw the eyes. I saw my hand with eyes on it.
Uh-huh. Everybody saw that? I doubt that it has worked for every. Yeah? Okay, I'm just going through the pictures with just seeing nods. Yes, yes, yes. No, no. No, I can't tell. Somebody says no, but I can't tell your name.
I haven't seen it. I'm not sure if that's just like the distance or the angle I'm doing it at.
Oops, what am I doing? Okay, everybody managed to. Sorry, did it work for you at the end of the day?
It
I haven't got
hasn't.
it either.
Okay, no. So I think let's. I was confused at first by the directions, but put your hand so that it's reflected in the mirror, right?
Yeah, you see your hand in the mirror.
Oh, okay.
You want to see your hand in the mirror.
Right.
And then, so your hand should be sort of like beside your head because you're trying to see it on the mirror. And then you want to move it around so that, you know, if it's right on the mirror, it should be on top of the monkey's face. If you're looking at the monkey's face straight ahead. And then you should see your hand go transparent. So like I'm sitting like this, and then I look at the screen.
Another thing is, Arash suggested using your left eye, but if you're right-handed,
shit,
maybe,
I am a lefty. Sorry, do it with your right hand. That's right.
I saw the face of the monkey blending into my hand.
Right.
So like a hand with a little monkey face. Oh, that was so bizarre.
Right. Yeah. So what is happening now? It is that image from one point in your visual field is going to one point in your cortex, right? But with this mirror trick, you're giving paradoxical images to your V1. One of the eyes is saying at that spot in the visual field, there is a monkey. The other eye is saying at that spot in the visual field, there is a hand, right? And the system should make sense of this discrepancy. What it does is that it suppresses one of them, and goes back and forth between them. Basically, everybody saw that? I doubt. Yeah, so I'll try.
I ended up seeing it.
Yay.
You saw it. Good.
So Arash, I can ask this during the stereo lecture next week if you prefer, but it's a little bit related to the idea that stereo vision might have evolved not necessarily for 3D vision, but for X-ray vision. and we can talk about it next week.
For what vision? For what
For
vision?
X-ray vision, the idea being that animals in more cluttered environments, it's not that useful to be able to see all the way around you if you can't see that
fair
far
enough.
in depth.
Right.
So seeing through like leaf litter, the idea is that if there's a lot of leaf litter in front of you, if one eye can, if the two eye images are a little bit different, you can suppress the leaf that's coming.
Interesting.
And Chengiz got a nice analysis of animals in environments with different degrees of clutter. But I'll bring it back up at stereo lecture.
Right. No, but this is very related actually to this topic. Yeah, that's a very good suggestion, because actually that helps with this problem, right? Basically, having that rivalry helps to have. I don't know. I find the word X-ray vision a little uncomfortable, like there's no X-ray going on there, but yeah, it's basically filling in the gaps of the visual field, and that could be one possibility, one serious possibility. Yeah. So so a few words about blind sight. I didn't put a lot of slides on the blind sight, and wow, we're running out of time. What happens if V1 gets damaged? If V1 gets damaged, you don't see, you go blind. If your V1 is gone, you're blind in the part of visual field that corresponds to the last V1. If all of V1 is gone, you don't see light anymore. Light is gone. Light is an experience that comes from activation of V1. Activation of V1 for your downstream means light. But what do you see? You have got other wires going on, right? Not everything goes to V1. There are these collateral paths that go to other cortices. There are these subcortical structures. So if you don't have V1, your eyes respond to light, your pupils respond to light. If something moves toward you, your pupils respond. You might even pull yourself away. If you're asked to post your hand through a slot, you would say, what slot? I don't see anything. And then you're like, yes, fine, but can you post this letter? You can actually do it. So there are some functions that are still intact, but those are very minimal functions, and they are not the main visual function. That is called blind sight. David Leopold has a very nice study on the physiology of blind sight, which I recommend to everybody. Another point that I want to mention about V1, which is a kind of funny property of V1, is that V1 is larger than the visual field. And what does that mean? What determines the boundaries of your visual field? Eyeball is a sphere, right? Yes. The boundaries of your orbit, right? The eyeball is in the orbit. So your eyebrows up there, your cheekbones down here, they block your vision, right? And they determine the frame of your visual field in each eye. But your retina is bigger than the visual field. Your retina is kind of, see this orange crescent here represents the retina. These black lines are the orbit bones, and the visual field is determined like that. This is mapped onto V1. But now, when you look downwards, the frame out there remains the same, right? So now a new part of your retina is exposed to light. And that is to cover the eye movements, basically, because your eye is behind an aperture, which is the frame of your skull, right, your orbit, and you want to move your eyes in there. So your retina is looser than the visual field, is bigger than the visual field, so that no matter where your eyes go, you don't get into a retina-less part of the visual field, right? And that means that there are, so when you go on V1, that becomes funny, because there are parts of the V1 that are not activated at a time when you look at the image. When you move your eyes, the image moves. The image gets those parts activated or not. So that part of the retina, this part of the retina, is now pointing to an impossible point into your nose, right, into your mouth. there is nothing there to be seen. And but you've got retina there, and there is an interesting debate on is your brain actually canceling out the signal that is coming from those parts or not. And now we want to do another experiment. This one I highly doubt that will work, because we need perfect light conditions, and I don't think we can recreate it. But let's give it a shot. Let's give it a shot. What I want you to do is to look at that red A on the top. You see V1 is larger than the visual field. There's a red A. Fixate at the red A. So what you're doing now, please keep your fixation. We want to do it for one whole minute. So what you're doing is that you're adapting your retina to that target shape pattern, right? And the part of your retina that is exposed to that target shape is far periphery down, right? Basically top of your retina on the bottom visual field. So what I want you to do after this, you will see an after effect. But I want you, after like another 30 seconds is passed, keep looking. I will ask you to close your eyes and roll your eyes. Your eyes are looking up now. Then roll your eyes all the way down and look with your eyes closed, look downwards. You might be able to catch a glimpse of that target after effect in your mouth. You might see it in your mouth, which is way outside your visual field. But you need to close your eyes and move your eyes down and wait for it to emerge. Ready? 1, 2, 3. Close your eyes, look down. Who sees it?
I see it.
It's a weird experience. It's like that target inside your nose or inside your mouth.
Yeah, I saw it.
I can see my whole computer screen. I can see the whole Zoom.
Exactly. Yeah, you see the whole frame. Exactly. The whole frame goes down into your mouth.
There was a
in my nose.
I was fixating on the A and the entire spiral disappeared.
So here is the interest. The reason there are two reasons I am not expecting all of you to see this. One is the experimental setting, that we need like a darker room and all that. You saw that too.
Arash,
I see smiles.
I found a trick that helped me to see it like very vividly. So I did not see it for a long time. So I took my glasses off and I covered my eyes. So I tried again and I was trying hard and I didn't see it. So I was sort of giving up and I took my hand and I saw it and I put it back and I started flickering and I started seeing it very vividly.
With your eyes closed?
Yeah.
Perfect.
But the trick was to put my hand, make it dark. And when I put the hand out and in, this like flicker made me see it.
Your hand with your closed eye?
Yeah.
I get that too, actually. Yeah.
What?
Because
I didn't follow.
It changes how much light is hitting his eyelids, right?
Oh, yeah, that's clever. Let me try.
It worked for me. Before I didn't see anything. But
yeah. Flicker should help. Yeah.
Yeah, and it came, and it was like not that it sort of came. I didn't see anything, and then I saw it very vividly.
I also found that the longer I stayed in that position, the more it began to crystallize and get brighter and more apparent. Like I look down, and if I continue to look down and hold that, it really starts.
Yeah. So if you want to see a vivid version of that, again, I can tell you what to do after the class. Get that flashlight that you're going to use for the shadow experiment on your eyes, and you can use the flashlight that like look up and shine the flashlight here. Hold it in your lower visual field, shine it directly to your eyes. That's a really strong, powerful stimulation. So you will get a very strong after effect. Then you close your eyes and roll your eyes down. That after effect will go into your mouth. Or if you do it the other way around, it goes into the top of your head. So anyway, the reason I brought that up is that it seems that there is a variation in individual. Dawan Wu and Patrick Cavanaugh have done these amazing experiments. We don't have time to go through them, but there is plenty of individual variation in there. Some people actually cut that space out. That impossible space that goes into your bones, actually they actively suppress it. They don't see it. Some do. So let's see, how are we doing on time? I am beginning to think maybe I should leave the rest of it for the next session, because a lot of stuff are still left. So let's be slow. Let's be slow. I'll just go over one more point and continue. We'll take our time and continue next week. I guess that's better than rushing through it. So the other thing that I want to talk about, basically we started, these were all mostly anatomical issues. So now I wanted to go into this issue of information processing in the primary visual cortex. How do we process information in the primary visual cortex? And that is a quite interesting topic. It starts from linear systems analysis. There is this idea of Fourier transform. This is a math idea. The basic idea is this. Imagine you've got a function. That's a function. That's a curve, right? You get that. The idea is that you can actually, you can show it mathematically that you can have a set of sine gratings, sinusoidal, simple periodic gratings. For every complex shape of every pattern, there exists a set of simple sine gratings that you can add to each other and you get that weird pattern. That's the idea of the Fourier transform. So basically, every waveform can be broken down into simple sine gratings. Even a waveform like a square wave with sharp boundaries, it is shown that if you get a sine wave and you sum it up with its odd harmonics, like go from F to 3F, 5F, 7F, and sum them up, it starts to shape a square wave. I will end the class today here, but I'm gonna send you this thing in the chat window, this link, and it would be nice if you can watch that for the next session, for the next meeting. That's a link to this YouTube channel called Smarter Every Day. Some of you might be familiar with that. He's explaining how does Fourier transform work, basically, in that. And I ask everybody to look at that and get familiar with the concept of a Fourier transform for the next meeting. And from next meeting, we'll talk about the actual information processing in V1.
Did you post it? I don't see it in the chat.
Oh, I thought I did.
I don't see it either.
Everyone in the waiting room.
Oh, you might have sent it to the waiting room.
Send it to the waiting room.
Oh man,
they can't come to the class, but they can watch this video.
I'm like the purgatory. You send
everyone. Yeah. That's right.
There we go.
Yeah. How about that?
Yep.
Yeah. So take your time, watch that video. And if you're more interested in the math, there is a lot of materials to follow, but he's explaining it in a very simple language, which is helpful. We don't have time to go over the math part of that here, but if you watch that would be helpful for us next time. And I guess here I end the session, and thank you everybody, and see if you have got questions.
So I have a question about the Retina topic map on V1 that we were talking about earlier. So it's something I thought that was interesting about it is that there's blood vessels represented and the optic disc as well. And it's weird because those are different. The optic disc, there's no photoreceptors below it. For blood vessels, there are photoreceptors below it, and that there's a illusion that you can observe called like blue field atopic phenomenon. If you look at a blue sky, you see these moving dots sometimes, and those are white blood cells moving around in those blood vessels. So you can see those. So I'm just curious. I mean, maybe I don't quite understand the imaging technique that was used in that paper, but why would that show up in the exact same way? Like you can see
Good
below
question.
those areas.
Good question. Good
I don't know if
question.
anybody understands that, because I don't really get the technique. I was looking at the paper and out of the corner of my eye, but I still don't understand it.
Yeah. Well, the reason you see the white blood cells and red blood cells is not because you see them through those blood vessels. Those blood vessels cast big shadows, and the photoreceptors under them, I doubt that they will make it. So they will be pretty similar to the optic disc. And the reason you see them when you shine light through the white of your eye is that the shadow moves to the neighboring ones. However, these are the big blood vessels. The blood vessels break down to teeny tiny ones, eventually to the capillaries. And capillaries are all over the place, right? Every point on the retina, if you put a pin, blood starts to ooze out. So that means that there are invisible, really teeny tiny capillaries traveling there. Those go directly over your photoreceptors, and those are the ones, because you can see your blood vessels even at the center of the eye, which is not there's no major blood vessel there. And for everybody, the experience that Simon is referring to, I highly recommend you guys to try it. I doubt we can replicate it with a monitor, because you need a uniform blue surface. Why blue? Because blue is absorbed by the blood. So blue casts the biggest blood shadow that's possible. If you have something red light goes through the red blood cells, but blue is blocked by the red blood cells. So the best of the blues for that purpose is the sky. So next time you are under the blue sky, just look at a cloudless patch of the sky, fixate there for a few seconds, then you will start seeing these little things moving in your entire visual field. And those are your own red blood cells. Those are your own RBCs. And you can actually estimate the size of your photoreceptor by looking at them, because red blood cells are standard 7 micron in diameter. That's like a standard measure, like a standard candle in histology when you see a red blood cell. The other thing is that there are blood diseases like sickle cell anemia that can be diagnosed that way. You actually see the blood cells have changed their shapes, and you go to doctor and say, hey, put my blood under microscope. The thing is that you don't need a microscope to see your own red blood cells, because they are traveling very close to your photoreceptors and casting shadows on those. That's a pretty amazing experience. I recommend to everybody to try. Just lie down under the sky and stare at the sky for some time. You'll see them. There's numerous little dots moving around.
So that's different from floaters then. I always thought that maybe I had.
It's different.
Floaters. It's different.
Yeah.
Okay.
Yeah.
Oh,
Floaters.
I had seen these guys, but I just had no idea that they were blood cells. I had always
They're your red blood
what
cells.
the hell they were, but I never crossed my mind that they were blood cells.
They're like little things that are moving.
They're sort of rounded.
I think it's terrible. I hate that I know about it.
Well, the thing is that once you see them, it's hard to undo it. You know, and it's hard to undo it for the brain as well, because they're moving. So their shadow is moving. So it's hard to computationally. It's not like the shadow of the blood vessels. And that's where the engineer should be charged, should be ticketed. That's a problem that cannot be solved easily. But yeah, you can see them. Floater is different, what Rosa mentions. Floater is this weird patterns that you see, like a spiral pattern floating here. Those are proteins in your vitreous, in the eyes, that have coagulated and created those shapes. But these are pretty uniform shapes, standard red blood cells that are all over the place.
Are there is there anything else you can diagnose by looking at the shape of those besides like sickle cell?
Yeah, if you see some objects flying around.
I just mean, like, is there any other thing that changes your red blood cell shape as an indicator of illness? Is there anything? I just want to know if I have a new tool to figure out when I'm,
Well, I don't think, I mean, the illnesses affect their size, their luminance. I don't think those can be detected by the naked eye. But anemia, maybe, extreme anemia. Maybe like if you have less of those, but I don't know. I'm not sure.
Cool.
Yeah,
but that might be right. Isn't it possible then that you might be able to detect some kind of diseases by looking at V1, even if you're not conscious of it? So
well, I have a friend who's Baktash Babadi, whose research is actually related to that. He thinks that schizophrenia is disease of cortex, and we are mostly studying its frontal cortex outcomes, right? The behavioral outcomes. My speech gets screwed up, and he's like, if it is a disease of cortex, maybe you can detect it in V1 too. Maybe it is changing some, and he has found some measures that some basic measurements have changed in these patients, and the idea is that his idea is that we know how does V1 work, so maybe we can model schizophrenia using that.
Sorry,
but yeah,
I didn't get the name.
His name is Baktash Babadi. He's at Harvard Psychiatry Department. I put his name here in the chat. But yeah, it's possible. It's an idea. It's a possibility.
I had a question about the spiral experiment that we did. You said that there was variation in the outcome within certain people. Do we know what causes the differences in experience?
Good question. We don't know, though. That idea started from Patrick himself. Patrick had this type of migraine that he was lucky. He got the migraine auras, this colorful sparks of light that you get before the migraine attacks, but he didn't have any headaches. So he was a lucky migraine case in that sense, and he would play with his auras. So that's how he noticed first that, because migraine auras come from V1 activity, the blood vessels in your V1, and then he saw this arc of colorful sparks on top of his fixation, and every time he looks up, they disappear. So he thought that, oh, they are going into this impossible space. They are going into his skull, and some active mechanism is actually taking it away because that shouldn't be there. And so that provoked this experiment. They did some experiments with the after images. They also did some experiments with actually at gluing a fiber optic to a lens, to a contact lens, and putting it on the retina. So now with a fiber optic, you can shine light to this part of my eye, like the extreme far away white part of the eye. And that actually goes into the impermissible space. So, and there is variation. Some people see that clearly, even if it is in the top of their skull or inside their mouth. Some people don't see it as soon as it sort of exits the normal visual field. And is there any active mechanism to do that? It's not studied. We don't know. We don't know. We have no idea. I don't know if I've said that or not. This might not be the most appropriate point to say that, because our knowledge of V1 cortex is among the best we have in neuroscience in different cortical areas. But let's face it, let's do it now. Actually, even now you can say that. I personally believe we live in dark ages in neuroscience. Basically, the number of questions that you have to ask to hit the margin of the boundaries of my knowledge and the literature's knowledge are very small. Like there's usually second good question, third good question, the answer is I don't know. And that's because we just don't have any theory. We don't have any theory of how is information integrated in the brain, is presented in the brain, how is it basically warped and changed until it creates and supports behavior. We just don't know. We don't know. And what I hope to achieve in this course is actually raise more questions. Not there is this standard version. You go like read Candel or I don't know textbooks. They tell you the way things are, right? But I personally hate learning science from textbooks because it is not about. It's not a religious text that you just follow and find the answers in there. It's the way you question about how the system works. And so what I like to communicate in this course is actually those cracks in our knowledge. Even in case of V1, there are lots of cracks. There are lots of things that are not done. Physiology of these experiments are not done. Like what happens if you record from part of V1 and the monkey looks outside or inside? We just don't know. And I think it's fun that we don't know because that's what you guys have to do. I mean, there's plenty of things to discover.
I wanted to say I didn't know that about Patrick with his auras, but David Hubel used to also get auras without the migraines, and he too would do these experiments with himself. Like I used to come into the office, and sometimes he'd be doing that simple blind spot test we were doing, and he was trying to see how his aura would interact with, when you get the filling in of the bars and the dots. And I think he actually ended up publishing something about it. I just tried to find a paper because it can be a useful tool because that activity starts in V1 and moves across. Like if you have an aura starting in your right hemisphere, when it moves across your visual field, it doesn't cross the midline, right? Because it can't jump across a gap.
Right.
So anyone who has auras, it is an opportunity to uncover something.
Yeah, I'm very jealous actually because I've never had auras, and I feel jealous. It's an interesting opportunity.
Yeah. So what he said he found is that when he's experiencing an aura, that if you're looking at those lines, the line passing through a scintillating scotoma does not get filled in. You get these clearly defined sharp gaps, which is interesting.
I see. Interesting. All right. Yeah. Okay. Thanks everybody, and we'll catch up next week with continuation of this.
Cool.